All right, welcome everybody. So today we are going to be demonstrating mitosis using a whiteboard, an expo marker, and some pipe cleaners. So first off, we are going to go ahead and draw our cell membrane. And even though this is a little bit square shaped, we are representing animal cell mitosis. So ignore the block shape. And we also have a pretty large nucleus in the center, and that's not to normal cell size scale, but we're just going to go with that so it's easier to focus on our subject areas. All right, so during interphase of the cell cycle, the cell is growing and it's getting ready to prepare for mitosis. And in the center of the nucleus, we have our chromatin, which is a bundled up mess of DNA. It's not really organized. It's kind of like a bowl of spaghetti noodles. So these look a little bit more organized than it would be. And it's kind of a jumbled mess. And we also have our centrioles that appear to prepare for cell mitosis. During interphase, our DNA actually replicates so that we can have identical DNA for our identical cell that we will copy. So now our DNA is copied, and actually it is ready to enter into prophase, the first stage of mitosis. During prophase, the nuclear envelope is actually going to start to dissolve. And our chromatin is going to bundle up with histones to form chromosomes. And again, it's not in an organized fashion. They're bundling up to form chromosomes. And now we can see it under the microscope, and the chromosomes are actually more organized so that we can have an easier time replicating the cell and DNA can go in the right spot. Everything can happen correctly. Okay, so now prophase. At the end of prophase, our nuclear envelope has completely disintegrated. It is all gone now. And our at the start of metaphase, which is the longest phase, and our centrioles are going to start migrating to opposite poles. So this centriole is going to come over here, and this centriole is going to migrate over here. And so during this time, it just takes a lot of time for everything to organize itself. The chromosomes are going to organize themselves in a fashion along the metaphase plate, which is a lot like the equator. It goes straight down. And once it has finally reached the metaphase plate, these meiotic spindle fibers are also starting to form, and they kind of look like stars called asters. So we call them asters. All right, and during metaphase, the spindle fibers are going to attach to the centromere, which is the center point of the chromosomes. And they're going to attach on either side so that both sides, both centrioles, will be able to pull on the DNA and have equal DNA on both sides. So again, the centrioles are holding on to the centromere, which is the center of the chromosomes. All right, now, once metaphase is complete, it'll move on to anaphase, which is where the centrioles use their spindle fibers and pull back and retract what are now the chromatids. And again, this would be easier because these spindle fibers attach directly to the centromere, so they'd be able to pull these apart really easily, faster than how I'm pulling them apart. All right, so each one of these pairs, a single one is called a chromatid. A pair is called sister chromatids. So these are all sister chromatids, the colors matching. That's what the colors are representing for you to see. And during anaphase, this would all happen kind of simultaneously, but the spindle fibers will pull the chromatids back towards their pole. So now we have our chromatids at either side of the cell, on either side of the poles, so that will prepare for cell division, the final parts. And now telophase is going to happen, which is our last of the phases, and our nuclear envelopes are going to start reappearing around our DNA. The centrioles will disappear, and they're split evenly on either side. And during telophase, we also have what we call the cleavage furrow. So if you think of it as somebody pinching down the middle, these are your actin and myosin filaments that are pushing these into two separate cells. And cytokinesis is happening, which is splitting up the cytoplasm between the cells, and also the organelles are shifting of where they need to be. And we now have our nuclear envelopes reformed, and we now have two exact daughter cells at the end of mitosis. And that is all.